So the first thing I want to do is start off with an introduction, if you will, to our computational resources we have. In particular, the AiMOS Supercomputer is the 26th fastest supercomputer in the world and the 5th most power efficient in the world. It's actually capable of performing over 8 quadrillion operations per second. That's an 8 with 15 zeros behind it in terms of math operations per second. To put that in perspective, that's the same amount of compute power as if every human in the world, all 8 billion people, were performing 1 million math operations per second, all at the same time. Now, this system was actually designed by IBM, and it serves as a testbed for the New York AI Hardware Center. This is a strategic partnership between IBM, New York State, Rensselaer, and several key industrial members. The center's goal is to design and test next generation hardware that will accelerate AI and machine learning tasks. However, in response to the COVID-19 pandemic, IBM, Rensselaer became founding members in the National COVID-19 HPC Consortium, which redirected vast U.S. supercomputing resources towards the goal of accelerating our understanding of the COVID-19 virus and the development of treatments and vaccines that will help address infections. To date, over 150,000 compute node hours on AiMOS have been allocated to COVID-19 projects from teams at the Cornell Clinic, Harvard, IBM, Northwestern, Rensselaer, and Cornell. Today, we highlight some of the groundbreaking computational work on AiMOS done by Professor Weinstein's group at the Cornell Medical College. Harel, I'll take it over from here. Thank you.
Thank you very much, Chris. To begin by telling you about that has become possible only with the use of AiMOS that Chris so ably described. I bring you back to Professor Hall's description, if you want, of what we know about the coronavirus or how we have visualized it in order to illustrate both what we knew and what we didn't know in pursuing our project. We knew that the ball that you see here in red contains the genetic material of the virus and has to enter the cell in order to release it. That is the mode of replication. We also knew that the skin of the ball must fuse with the cell membrane, but we didn't know how. We knew that the spike attacks the cell to allow the ball to enter, and we knew that it's a specific part of the spike that gets to pierce the membrane, but we didn't know how. And that is what our project went to find out and to use in fighting the entrance of the virus into the cell. So very briefly, you see here an enhanced view of that ball and spike that tells you how these spikes are used to connect with the human cell. What you see here is that the virus has evolved to identify something that exists in the human cell, a cell receptor called ACE2, and in its structure of the spike has created these receptor binding domains shown here in cyan, which attach to this cell receptor and brings the virus very close to the membrane, but not close enough to pierce it, not close enough to connect with it. And this was one of the greatest surprises in the scientific community, is how the virus manages to send fusion agents that allow it to insert into the cell membrane. What you see here is that the virus has done something extraordinarily clever in evolving to infect us, and that is to hijack an enzyme that is right close to that cell receptor. And what this enzyme does is to cleave the spike in one particular place, just one snip at this particular place called S2. And what this does is described here from detailed structure and massive computational work that we did on AiMOS to find out what is happening between the time that the enzyme cleavage has not happened and the entrance of the virus. So you see here that this fusion agent is closely packed to the structure of the virus. But what you see here is that immediately after enzyme cleavage, this fusion agents, the fusion peptide, begins to separate from the structure. And that separation triggers an extraordinary rearrangement of the virus, where this entire part moves down, as it were, from this place and positions these fusion agents as darts to being ready to penetrate the membrane. Because we found this out, we were able to use the computer in the really massive simulations to find out exactly the main mode of insertion of this fusion agent into the membrane, what it looks like there, and what it does to the membrane. These are extraordinarily important elements in our ability to move to the next step, which is where the project is right now with some very interesting advances in results. And that is again feasible only in the collaboration of our design work with the computation on AiMOS, because we are using artificial intelligence and the computational modeling, which we call simulations, because it goes through a what-if scenario, as I illustrated to you in the previous slides that Professor Hall mentioned, to discover compounds that stop the fusion peptides of the spike right here from entering. That is possible only because we know what the spike looks like when these fusion peptides have penetrated the membrane. And this information is shared with the group of Dr. Payal Das at IBM, and the novel artificial intelligence framework that she has developed, which combined with our work, allow us to identify a whole series of completely new kinds of compounds that will stop and that can and do stop the spike from entering the cell. And this is where we are now for the benefit of everybody. And I hope you wish us luck. Thank you very much.